Hi, Monique. Thanks so much for joining me today. I really appreciate it.
Oh, thank you. Thank you for receiving me, to having me in your podcast. I'm very happy to be here.
You've worked across pathology, environmental chemistry, metrology, and biological imaging. What originally drew you to analytical chemistry, and what has kept you there over such a diverse career?
For me, the answer is clear now that I have experience in the area, but that's basically evidence over opinion. What that means, using analytical chemistry, you can use tools and methods to achieve more accurate values. So one example is the calibration curve that's going to give you the value that's close to the true value we are interested. That's very, very important in all these areas you mentioned. So if your method is solid, the sample is going to tell you the truth. That's basically that. So in the end of the day, it's all about trust, the value you work with, and thinking about which that result is telling you. So, and I like this responsibility of making results reliable.
I think that's a great answer. You trained and worked in Brazil for over a decade before moving to Australia. How did that transition shape the way you think about analytical science and its role in public health?
So in Brazil, we have big contrast between the two countries. In Brazil, I had about 10 years, as I said, experience working in different areas. And I learned about resilience. That was the main thing. It's a rich country with a very low investment in science. So that means you need to make things happen even when you have the access is limited. So for me, the mindset of quality is the same between both countries. The difference I see is the access. In Australia, I have access for new instruments. I don't need to be counting the standards. I have CRMs. I have access basically. And also, it's easier when you try to fix instruments. Of course, you have limitations in Australia as well, but in Brazil, I feel that's more, how can I say, it's complicated to get things done. So that's made me learn how to do things and be patient as well. So I learned how to fix instruments, help the engineer, try to minimize the time between fixing and buying pieces of instruments, work with different methods. Sometimes you can buy a CRM, but you can produce one standard to check your method. So yeah, I was very resilient. In Australia, I have more consistency in access these results, what let me focus more on the consistency of my method, the refinement of my method, and escalate as well. So go further when I'm working like specific project. So the matter for public health, because reliable results deliver faster, can support better decisions. So can impact our patients' life and community in general.
Metrology runs like a thread through much of your work. Why do you think metrology is still underappreciated by many scientists outside of standards and calibration labs?
Yes, metrology is it's often overlooked because when it works, you don't notice. So you only notice when something goes wrong. So I think I just have few points for metrology. That's my personal opinion. Is the incentive is mismatch. So it's when the most scientists are reward for novel results, not for providing measurements or guarantee the measurements are traceable, comparable, and defensible. So you produce the values and you take conclusions on it. There are second things the black box instrument culture. So the modern instruments feel like plug and play. So people assume the number is true because of the software just produce it. So when you don't have a deep knowledge of validation and metrology itself, is can happen. So also I believe that's the confusion between precision and accuracy. So most people see nice repeatability and think that that's equals correct value. So that's two different approaches that need to be clear as well for the scientist. Then they can take decisions what he needs to do each step. Another thing is uncertainty feels abstract and so hard. So when you read, you just open uncertainty book is just blown your mind. You can't keep going. So I think we need more clear approach for this. So measuring certainty and traceability chance and uncertainty budgets can sound like statistics instead of science. If you can translate it to science language, it's going to be probably more used for scientists. I understand how hard is this topic. The another thing is the time pressure and the cost. CRM, when you work with replicable runs, control charts, method validation, takes time and depend on the time you have for your project. That's the first thing that's not investigated. And as I said, the education gap. So many degrees teach how the analysis and method steps works, but just talk very, very light on measurement and metrology. I believe that if a more clear and inclusive approach for metrology would be useful for scientists in general.
In your experience, where do analytical methods most commonly fail in terms of traceability or uncertainty? It could be sample prep, instrumentation, data interpretation, or something.
Yes, I see that the most common fail, that's what I always talk with my students. If I would choose one, I would say sample prep. The most failure start in sample prep. So we can attribute this to contamination, like dirty tubes, lab air, lab reagents carry over. The second one is recovery, so it's not studying the adsorption for the sample, volatilization, filtration, matrix effects, poor representativeness of your sample, so sampling, heterogeneity, and missing or having like weak controls. This is going to impact the traceability uncertainty because change the sample. So, and the error can follow the whole chain. So it's going to be impact whole the process. And then other two as important as well as the data interpretation. So when you need to know like about calibration curve, LOQ, LOD, linearity, can't ignoring the drifts. That's very important. So this require a knowledge and not just in the to know what is, but how to do this during the process, how to access. And the third one is instrumentation. So that could be the drifts, carryover, detector saturation, an alignment. So I would say these are three: sample prep, data interpretation, instrumentation, but 99% is attributed to sample preparation.
For early career researchers, what are some of the most important habits they can develop to ensure their data is genuinely defensible and reproducible?
That's a very good question. So I have 9 points, 8 points, sorry, that usually I use to show my students I like to approach this topic since when I start teaching. So first thing is write things down as you can. So have one lab notebook, date, the day you're doing your experiment, number the page, and then you know the sequence, how that works. And in case something goes wrong, you can trace back and find where is the problem. So when you don't write things down, it's very difficult for you to organize and keep going with your research or your studies. So second one would be consistent with sample prep. So be aware of cleaning, how to prepare your sample, go to literature review, check what people are doing, what you need to be done for that specific sample. And also regarding to the instrument you'll be working with. So we have different types of sample prep for different instruments. So it's good to be aware of what can be done. So always run basic controls, blanks, instrument blanks, duplicate spikes, check standards, CRM as well when it's available. Sometimes the place you're working in is not like investing in CRMs, but you can also prepare standards. So if you're preparing your calibration curve with one specific standard for one brand, you can use another brand to cross-check how is your calibration curve behaving. So also check for QCs before trusting results. So don't analyze your sample before you check all this calibration drifts, recovery, and carryover. So this is the quick check. You need always to go back and check before you analyze your sample. In terms of sample, another thing that I think is very, very important is check concentration. Sometimes better to start with diluting your sample to find the proper concentration for that specific instrument. Then you analyze a sample with high concentration, saturate the instrument. So it's going to be a big job of stop everything, clean everything, try to drop all the values. So this happens a lot in our ICP-MS. The sixth point is keep raw data safe. It's very important to have your data named with dates and do backups. It's very, very important. So repeat properly. So check repetitivity and reproducibility as well. And don't reuse calibration curve and sample. So just re-prepare, don't re-inject. Always preparing fresh. And be honest about the limit of quantification, detection. If it's possible, calculate the uncertainty. Try to use the parameters, statistical parameters like standard deviation, relative standard deviation, to access your data and see if it's reliable, is what you're expecting, and always the calibration curve. If you document well, you run your controls, review your QC every time, your data becomes strong and it's something that people can trust and you can keep going with your research.
I think that's a great transition. And one thing I'd ask you to do too, if you could, I think this is the first time on the podcast we've discussed LA-ICP-MS. So if you could get into a little bit of a overview of what that is, but your PhD work used multiplexed LA-ICP-MS to study muscular dystrophy. For listeners who are less familiar with the technique, what makes it such a powerful tool for biological imaging?
The turning point of LAICPMS is that you can ablate your sample in integrity without destroying, so you don't need to prepare liquids for the laser, so it's going to be ablating it. And the ICP is going to be quantifying it. So basically, in my research, I analyzed the glycoproteins, and glycoproteins are interacting all the time in a cell. When you use the conventional ICP-MS, usually you need a liquid sample, what means that the proteins can be destroyed during the digestion. With the laser ablation ICP-MS, you can analyze the tissue directly and see the spatial arrangement of those glycoproteins in place. Almost like take a snapshot of what they sit and interact in the muscle. And when you use a multiplex approach, you can not only co-localize in the muscle where it is, you can also quantify them at the same time. So if you have the glycoproteins interacting, it's okay for you to quantify and try to see the localization individually, but to be close to the reality, it's important to have all of the max of glycoproteins interacting at the same time and analyze it at the same time. So the laser is going to provide the spatial image, and the ICP is going to provide the quantification. So, and we use the gelatin-based calibration curve because gelatin is the closest match for muscle matrix and is accepted by the scientific community.
And I think you just touched on this, but feel free if you had anything else to add. My understanding is that traditional pathology often focuses on single markers. So, what does this multiplexed elemental imaging allow researchers to see that they would otherwise miss?
So as I said, the multiplex element images show the spatial relationship and the heterogeneity of many markers at once. revealing the partners, colocalization, regional difference across the tissue, and the single marker approach or average measurements would miss something. So it's not interacting at the same time. You need to watch when they are interacting. So while also allowing quantification, the same run, so you can have your snapshot, the image, then you'll be quantifying it. That's incredible. So I have story like during my PhD, we have quantify the monoplex was one glycoprotein per time, and then we did for plex. And we had a huge difference between both. What that means, if you trust the monoplex value, you can give like a wrong protocol for patient because it's not representing what's going on in the cell. When you have all the glycoproteins relating in the cell, you are looking at only one, so you don't know what's going on when they are interacting. Only one. That's mainly the difference.
What were the biggest analytical challenges in validating multi-element imaging methods for biological tissues?
So yeah, we have some challenge, and I'm bringing two. So the first one is the static hindrance. Static hindrance is when you have the overlapping. What means that when you target that specific glycoprotein, one could be overlapping the other and giving us a not true value for that specific glycoprotein per time. So in the cell, it's not a flat environment. So the glycoproteins are interacting and you don't know what's going on there. So when you have that snapshot, you don't know in which position they will be. And also, you don't know if it is going to repeat next time. It's a live cell. So we can't predict. So when you multiplex, they can physically overlap and block binding. So the signal can look lower, even if the target is there. So to overcome that, we reduce crowding by optimizing antibody concentration. So the most special close targets are in competing with the same mix. And the another thing was that what I mentioned previously, I was validating singleplex versus multiplex to check for signal validating suppression. So if it's needed, that's what I did in the beginning of my PhD project was I split the panel in smaller targets. Instead of analyzing 11 targets in once, we will start analyzing 3 and 2, and then we went increasing just because we know when they are the only 3 interacting, the chance of overlap is minimal. When you increase, if something happens, you know that could be due to the static hindrance. So basically, that's what we did to overcome this situation. We start with small panels, and then we went increasing this panel to see what was going on with 11 glycoproteins interacting at the same time. The signal is going to be reflecting the biology, but minimizing the competition during the hindrance.
And speaking of tissue, I know you looked at brain tissue using this same approach in Alzheimer's disease research. What insights can elemental mapping provide that complement biochemical or genetic approaches?
So when we analyze the metals or lanthanides by laser ablation ICP-MS, I mainly analyze lanthanides for DMD, and for Alzheimer, we analyze the essential metals. So how the main thing was not only quantify it, but understand how these essential metals were distributed in the tissue. I can give one example that was zinc, that's essential metal in the brain. In Alzheimer, it appears in cluster in certain areas, such as around plaques, rather being evenly distributed. So a total amount of zinc doesn't tell us anything about the disease, would be the same amount of zinc, but the colocalization is going to give us the answer we need to trace the patient disease evolution or maybe helping define the accurate treatment protocol. because you know that when in the health tissue you having the distribution equivalent, and then you can be tracing for the disease that's going to be changing, so it's going to give us more accurate. So we just was studying the dystrophin that's responsible for the disease. So how the changes happen in the muscle tissue is going to tell us the story of the development of the disease.
And much of your earlier and ongoing work involves environmental chemistry and metals, like you just mentioned, and complex matrices. What analytical lessons from environmental monitoring carry over into biological and medical research?
So environmental monitoring teaching us to treat complex samples seriously, matrix effects, sampling quality, contamination control, strong QCs, what makes results defensible. The same habits carry direct into biological and medical research, where you also need to think in terms of uncertainty and decision making, not just a number. So, for example, a calibration curve isn't valid just because it's linear. Linearity shows consistency, but CRM or standards proves accuracy. So your method can measure a known value correctly, and you can trust your results.
Early, you talked about how sample prep can be a common source of failures in general, and that's the most prevalent example, I suppose, of the failure really occurring before the sample even gets to the instrumentation. In your view, what are some of the most underestimated or maybe overlooked sample prep pitfalls in this type of analysis?
Yes, so in my opinion, the three most common and important sample prep pitfalls are: first, number one, contamination. This could happen from reagents, water, dust, gloves, benches, tools, containers. So ICP-MS is one big example of it. So easy to contaminate, can come from anywhere. Second, I would say inappropriate or poorly clean labwares. So each instrument requires different type of discontamination most of the time, when it's not possible to buy a new and free from the analyte you'll be using for that specific analysis. So the wrong or not acid wasted, solvents in not the appropriate degree for that specific analysis, or anything that can leach or carry metals or the analyte you are interested in your sample. And I would say the third one is the subsampling and heterogeneity. So a small a liquid that doesn't truly represent the whole sample could affect your final results. So if you control these three, I think you prevent most trace metals failures before your sample goes to the instrument. Principally the subsampling. So you should have the mapping, understand about subsampling. To avoid this is more used for on viral samples. It's good to understand mapping to do the procedure properly before you bring a sample to the lab and start preparing it. But you can include another dilution, or when people don't use blanks or don't check the system for blanks as well, or digestion extraction if you see that's mainly focusing the sample.
What advice would you give to labs who typically heavily invest in instrumentation but maybe underinvest in sample prep optimization?
Yeah, this is a philosophy that I believe the companies should have is strict sample prep like part of the instrument, because it controls what the instrument is going to be allowed to measure. So I would say spending money and time in sample prep, this including developing methods and also training people to do so. If it's not developing, at least to check time to time, principally in the companies that are ISO accredited. They need to have this audit time to time. It's good to disseminate this knowledge in for between all the the employees. Always run the basics. That's what I'm always keeping saying. Checking blanks, duplicates, spikes, check, CRN, standards, QCs. Drifts for the instrument, they are very sensitive for electricity. It can vary during the day. The instruments, they're analyzing 24 by 7. So this is very, very important to be checking and keep it consistent, checking the SOPs to follow properly. Same approach every time, check-ins as well during the day, and track blanks and recoveries. That sometimes can happen an issue, let's say drifts, but when you see that's the cross contamination, we need to stop and clean the instrument. This happen when a lot when you work with unviral samples in ICP-MS. So depend on the sample, you need to stop twice or 3 times a day to clean it. So it's good always to keep it consistent.
You've spoken candidly about how lack of long-term institutional continuity can interrupt promising research. How common is this problem, and what impact does it have on science?
Yeah, this is a very trick issue that's happened a lot, and I believe that's not only in Australia but in different countries where the investments in science are cut. And I always, when I read the news, I see this happening. So, but let's go for the science about what we're talking about. That's very common because people and funding often change every few years. We have time for the projects to finish. When these key people leave, a lot of knowledge leaves you. So most of the time, the train is not done, the knowledge is not disseminated for new people. I have many different projects going on in the same institutions. I don't blame the scientists, so we don't have time to be learning everything we want to. So files get scattered, methods are not fully writing down, and the next person has to start again. That's one of the issues I see that's happening. And this is going to impact science drastically because slow down the progress, waste money, make results harder to repeat many times. And instead of building on what was done, lab spending time rebuilding the same work. And when the continuity is lost, the impact can be severe. Specific projects sometimes are not finished, just not giving continuity. So the most qualified professional left and wasn't not worth of keeping with that project going on. Should be like wasting a lot of time and money investing someone else to keep going with that specific project. It's gonna impact everyone, not only the research itself, but society and community like that usually are expecting results from us to help like very important issues we have.
No matter what type of work you're doing, you never want to have to repeat it. Better handoff or better planning or both, better documentation maybe could have saved having to do that work a second time, I
Yeah.
suppose.
So in a private companies, you find that where you have a quality system, usually you have all documented. But for example, in universities, you don't see this. So I had all my research documented, all data organized, saved in OneDrive, shared with my supervisor, and I submit my thesis by publication, so all my data was published. So I had all in papers. I don't even need the data anymore, like save it just in case I need to prove something. But my research was all published. But I was already experienced in the field, so it was easier for me to do this. But most of the students they come from the honors or masters for the PhD. They are young. They don't know how to do this, how to document, how to organize. So that's not blaming them. That's very difficult to do at the beginning. Sometimes we have very good projects going on, but when you don't have anything documented. I believe if you have more supervision on this part as well of documentation, it's going to be very helpful. But most of the time, that's not a rule. They suggest, but that's something that's not a rule. But in private institutions, usually we have all documented.
For students or early career scientists who are listening or watching this, what mindset should they adopt if they want their analytical work to truly matter?
So my suggestion would be, I have like few suggestions here, would be resilience. That's first thing. Keep going when things fail. That's normal in real lab work. That's one of my suggestions every time when I start a new class at uni, because I know how frustrating it is when you try to do something and in the lab sometimes doesn't work. Number results are results. So instead of being frustrated, just enjoy the process. The second one, I would say critical thinking. Don't just follow steps. Understand what the step is doing, what they mean. That's very important. Not only to for knowledge, but to understand the next steps and decide that as well. Ask why. That's the third one. My suggestion. Why am I doing this? What could I go wrong? What decision will this data support? Where this data is bringing me to? So that's very important to understand the process as well. The fourth one, I'd say keep improving, learning, adjust, continuing, consistent over perfection. That's, I think, is a bit close to resilience. So the next one would be think impact. Who will use this data? What decision could it influence? This is very important as well to find gaps in research. So even to apply sometimes scholarship or try grants, that's very useful. So who is going to be benefiting from my data? Who will be helping? So this is a good question to do for yourself every time it's possible. And respect the boring work. So sample prep, QCs, blanks, documentation, that what we're talking about. Even if you don't know how to do it, just write a step by step and document what you're doing. Is going to be easier for you to trace and track what could be improved or what could be doing different. So, and then after, can imagine after 4 years, you're doing your project, you can't remember what you did in the first year. So it's very important to document it and be clear and honest. So know the limitations of your method and keep improving, keep moving.
I think that's great advice. Monique, was there anything else you wanted to talk about today regarding your work that we haven't covered or maybe we skipped over?
I think we spoke about a lot of my PhD project. A very tricky and sometimes complex topic to talk, but I have my papers very easy to find on Google Scholar. Typing my name and my surname is very easy and reading about it if you're interested. But yeah, just enjoy the analytical life. Sometimes you have frustrations, but so enjoyable. I really like the field and the instruments and see how we can impact and help people's life. So I'm very proud of being part of this, being scientist.
Very good. Well, Monique, thank you so much for your time. This will be a great episode, and it's been really informative to learn about a type of analysis we haven't covered that much, and obviously it's critically important work as far as Alzheimer's disease and other applications as well. So thank you so much for sharing with the audience.
Thanks for having me. And yeah, I'm happy to, if someone have any question, I'm happy to clarify and explain. Thanks so much.